So thanks everyone. So our third speaker is Bobbie Anne Turner from the University of Liverpool, and she's going to be talking about the investigation of the cellular interactome of the nucleocapsid protein of seasonal and highly pathogenic human coronaviruses. Thank you, Bobbie.
Hello everyone, my name is Bobbie. I'd like to thank the UK-ICN for giving me the opportunity to present my ongoing PhD project. And I'm based in the Hiscox lab at the University of Liverpool. So just a little overcap on the nucleocapsid protein. It is located at the 3' end of the genome, and during viral replication is the most abundantly expressed viral protein. It has a multifunctional role, which includes genome packing and encapsulation, as well as regulation of viral replication, transcription, viral assembly, among others. It's also thought to modulate cellular processes and recruit cellular factors to promote viral replication. And as such, it is a potential antiviral target. So the aim of my work is to investigate the nucleocapsid interactome of different human coronaviruses and compare and contrast them. So in particular, I'm looking at 2 alpha coronaviruses, so human coronavirus 229E and NL63, and 2 beta coronaviruses, OC43 and SARS-CoV-2. So I've got 3 seasonals and 1 highly pathogenic coronavirus. And the aim is to identify potential unique interactors, those that may influence disease pathogenesis, for example, and also to identify pan-coronavirus interactors, which may provide to be therapeutic targets. So in order to do this work, I'm looking at the localization and co-localization of Ns and their host factors, as well as doing co-IP, a methodology to identify potential host interactors, and then this work will be followed with functional analysis to understand and investigate the impact of these host viral protein-protein interactions. So my workflow, I've designed 4 constructs to express the nucleocapsid proteins as a GFP-N fusion protein, which are then transfected into 293 T cells, supernatants then collected for my downstream port-down analysis. So in particular, I use the GFP bead trap methodology, and this is where the GFP beads bind to the GFP-N fusion proteins, which then works as a bait to pour down my cellular interactors, those that are bound or interacting with N. And then my eluted samples then run through shotgun DDA mass spectrometry, and the mass spec work in particular was done by a postdoc in the lab, Stu. And then, so Stu's put together this lovely Venn diagram. So the data was analysed to meet quality thresholds, and from that we found over 200 interactors that interact with all the 4 different nucleocapsid proteins. And this Venn diagram just shows the distribution of shared and unique interactors. So SARS-CoV-2 had 125 interactors identified, so this sort of bluey, purpley ring here, of which 4 were unique to SARS-2. OC43 had 137 interactors, of which 6 were unique. NL63 saw 178 interactors, of which 8 of those are unique. And 229E, we saw the largest number of interactors in this data set at 212, with 36 of those unique. So it also had the greatest number of unique interactors. And also we saw a larger overlap between 229E and NL63, especially when compared to OC43 and SARS-CoV-2. And in the center here, we have the number of shared interactors that were found across all 4 nucleocapsids, which was a total of 104 proteins, which equated to about 44% of the total number of interactors identified. So I looked at the molecular function used in Panther of the 104 shared interactors, and there was two main prominent functions between them. So essentially these are the ribosomal proteins, structural constituents of ribosome, and then one of the main functions was binding. So of the 104 proteins, about 64 of them have a role in binding, and of those 64, about 90% of those, 60 of those, have a role in RNA binding specifically. Now, as we know, N also binds with viral gene RNA, so it's not a surprise that a lot of the host interactors are also RNA binding as well. We then use string and cytoscope software to analyze the unique interactors and try to group them to identify functions also. So NL63 and 229E had a large proportion of their unique interactors consisting of mitochondrial ribosomal proteins. And the solid lines here are the ones that the interactions identified in this study, and the dotted lines are all the interactions that were identified in string. So there's a possibility that they might form a complex together. For 229E, we also saw GSK3 alpha and beta solely in that data set. I found this interesting as GSK3 alpha beta has been documented to phosphorylate the nucleocapsid proteins and has literature specifically for SARS-CoV-2N. So I'm curious to see why I've only got it in my 229E dataset. So those are my alphas. I have included my work on my beta coronaviruses as well. I know it's an Alphacoronavirus section, but it's only half my story otherwise. So for OC43, 4 out of the 6 unique interactors identified were part of this 1433 protein family, which all has a role in intracellular protein transport. And reassuringly for me, in SARS-CoV-2, I saw the stress granule proteins G3, BP1, and 2. If you've looked at any nucleocapsid work for SARS-CoV-2, these proteins have been heavily documented to interact with SARS-2. So it was reassuring to also see it in my data set and to sort of confirms that my methodology is accurate or works. So these are a list of the proteins that I've chosen to down select at the initial stage. So 3 of these at the top here are some of my unique interactors. As I mentioned, G3, BP1, and 2, heavily documented, so I'm almost using it as a control at this stage moving forward. And the bottom 5 here are some of the unique interactors that I've identified. Now, there are 104 in total to look at, so to filter these, I ordered them by fold change when compared to the control group, and also looking at their abundance as a way to measure interaction strength, and filtered those greatest to largest. And consistently, these were all in the top 10 to 15 for all 4 of my nucleocapsid proteins. So it gave me a solid base to go off that these were high coincidence interactors. A lot of these have also been documented to interact with other nucleocapsid proteins from other viruses, so nucleolin here with IBV, as well as some of the other porcine coronaviruses, the same with DHX9. Additionally, ILF3, DHX9, and YBX1's also been reported to interact with the NS1 protein for influenza as well. So again, just building that base that these are solid interactions. So to validate and confirm these bindings, I use reverse co-IP methods. So this is where I use a host antibody to the host proteins as my bait, and if successfully bound with N, it will pour down my N in my samples. So my eluted samples are then run on a Western blot, lovely Western blots, and I probe against 229EN or NL63N in this case, and if my pour down's been successful, I should see my N in my sample. So whilst I do find presence of N, and I will just say these are again using my GFP-N fusion protein, so the molecular weight is around about 70 to 80 depending on the construct. I am detecting N in lanes where I wasn't necessarily expecting it. So ongoing, it is I need to do further validation to determine why I'm detecting it in samples. And I've also included my betas again, and again, I'm getting the reoccurring issues or challenges of where I'm seeing it in lanes where I'm not necessarily expecting it. So as well as continuing on with this, I'm also looking at my functional assays to over and underexpress protein of interest, and this is done using siRNA knockdown methodologies to underexpress these host proteins, and then using constructs to express my host proteins to overexpress during viral infection with an aim to understand their impacts on viral biology. So I'll finish there. Once again, big shout out to Stu for his help with the mass spec, and a big shout out for the rest of the lab for all their moral support in this journey. And thank you.
Hiya, great talk.
Thank you.
While you were preparing your samples for mass spec,
Yes.
were they done in the context of infection, or were they just transfected with that GFP construct?
It was just transfection for mass spec into 293 T cells. And I'll do virus infections with like the siRNA knockdowns and stuff like that.
Fail. With the RNA binding proteins that you pulled down, a lot of those, do you
Yes.
think you're co-precipitating those with host RNA?
Yes, so I did mean to say, for my reverse co-IPs, because of the RNA binding activity of my host cells and the ends, they were RNA treated to remove the RNA and then to hopefully eliminate those non-specific RNA binding interactions. So again, that's why I'm a little bit like, why are they there? So yeah, thank you.
Sorry. Yeah, it was a similar question. I think you could do those co-IPs with and without RNAs, right? That might help you pick out what's going on there.
Right.
But I had another question, which was, I think for SARS-CoV-2N, I mean, that's the only one I know a little bit about. I know there are alternative transcripts maybe of N. I don't know if they share any of the homology, like N star, I think.
Right.
Maybe has innate immune modulatory effects. I don't know if you've considered any of the other truncations of N.
No, I haven't.
For the IP, I don't know.
Yeah, that's a great idea. Thank you.
Right. How far are you allowed to throw this before you get in?
Go for it. I've got my question now, you put me on. No, I've forgotten it.
Okay, seems like everybody survived this. All right, so thank you so much.
Thank you.